Okay, so here is our DNA strand. Now remember, we talked about DNA is a double helix, which means we're going to have two parallel lines here in a second. And DNA is also anti-parallel. We're going to see these going in this direction. We are going to build our complementary strand going in the opposite direction. So we're going to start off with this one, A. This stands for adenine. And remember, apples grow on trees. So our complementary base pair is thiamine right here. Our next one is thiamine, and so apples grow on trees. Here we have our complementary base pair right here. Next, we have guanine. Our base pair complementary is cytosine. Gas goes in cars, or we could say cars go in a garage. And then we're going to see C pairs with G. So we're building our complementary base strand. Notice how these are going in this direction, and this one is going in this. There's a reason for this. The reason why we call it 3 to 5 prime is based on the carbon bonding. And this is going to go here, here, and we've officially finished building our DNA strand. Our helicase is our unzipping enzyme. Anything ending in ASE, 99% of the time it's going to be an enzyme. A helicase is going to help us to unwind our double helix. This is a double helix, helicase, okay? Now remember, we talked about DNA is anti-parallel. We have one strand that goes in the 3 to 5 prime direction, and this is how they count the carbons in our sugar phos backbone. We have 5 to 3 prime, so anti-parallel. We are going to put DNA through our helicase. This is how we start off DNA replication. So our helicase is going to unwind or split open our DNA. DNA polymerase is another enzyme that's involved in DNA replication. And remember, it ends in ASE, this is an enzyme. Poly means many. So DNA polymerase is going to essentially build many of our nitrogenous bases. It's very important in this step to notate that DNA polymerase is going in this direction, 3 to 5 prime, and this one is going in the 5 to 3 direction, which means as we are building, one of them is going to be the leading strand because this is just going to be able to continuously keep going down. Our lagging strand is moving in this direction, which means it's going to have to keep jumping backwards to catch the new strands. So this is our lagging strand. I'm going to make our copy in gray. So we just added a thiamine. Apples grow on trees, so now we're adding an adenine right here. Then we're going to add a cytosine. We're going to add a guanine. The reason why we're making this in gray is I want to highlight that DNA replication is a semi-conservative process. Semi-conservative, which means we're holding on to 50% of the old strand while creating 50% new. So semi-conservative, one old, one new strand. This is also happening with DNA polymerase up top. So now we've moved further along with our DNA polymerase, and this is what I meant by the lagging strand. Remember, this is going this direction. So now we're going to have to jump backwards to grab onto it. And so this is actually going to be made in fragments, and these fragments are called Okazaki fragments. Ligase is going to have to come in and glue these fragments together. Meanwhile, our continuous strand is just being built. When we're finally done with DNA replication, we end up with 2 daughter strands. Remember, semi-conservative. So we see one old strand, one new strand. So we have 2 full daughter strands.